Hey guys, welcome to today's lecture. Just before we get started here talking about enzymes and redox, just a brief reminder that we will be writing our class test next week, Tuesday. Please be sure that you are prepared and ready for it. This will be the final lecture that you will be included in the testing material. So being sure that you know all 8 of these lectures and the content they cover, including the material in your book. And yeah, if you study and prepare, you will be fine. So don't worry too much. And I hope you guys prepare during the weekend. Okay, so today we'll be talking about metabolism, enzymes, and redox, as the slides indicated. So the term metabolism refers to the sum of all chemical reactions within a living organism. And as chemical reactions can either release or require energy, metabolism can be seen as an energy balancing act. Consequently, metabolism can be divided into 2 classes. One, you can have catabolism, which is the degradation of compounds with the goal of energy production. So here you can see a large molecule being broken down into smaller components and producing energy. And you also have anabolism, which is almost, as like I said, we can think of it as the reverse. This is a biosynthesis of compounds. For example, you can think of this as a protein. So if this is a protein, this will be the amino acids, the small molecules that make it up. And you will be investing energy, or the microbe will be investing energy to synthesize the larger molecule. And these enzymes are important during both of these processes for them to function properly. You can then, as I briefly mentioned before, you can think of catabolism and anabolism as 2 inverse processes, sort of like flip sides to the same coin. So essentially, when a complex molecule, something like glucose, is split apart, the energy is transformed and trapped into ATP. So here you will have a complex molecule, a glucose, for example, that's broken down by catabolism, will result in some heat being released, waste being expelled, and then ATP being produced, which is how your body stores or how biological organisms store energy. And then in the smaller molecules, these smaller molecules will then be used as building blocks during anabolism, and so too will the energy. Thus, the catabolic reaction can kind of be thought of as providing the building blocks for anabolic reactions, which will then result in the synthesis of larger molecules, such as your ribosomes or your cell wall and any other thing the cell might need. So the reactants, products, and intermediates of enzymatic reactions are known as metabolites, which are modified by a sequence of chemical reaction catalyzed by enzymes. I'd like to just draw your attention here to this background image. This is a small portion of the interactive metabolic pathway map created by Sigma Aldrich. I'd strongly encourage you guys to go look it up and download the entire thing. I think there are also posters that they sell of these, if I'm not mistaken. This is really impressive because it gives you an idea of just how insane this metabolic, how many metabolic pathways there are and how they are interconnected and interwoven. This will really help you to further your understanding, and especially as you continue your studies further in biology. But you do not need to know these all these pathways for your exams or for this course at all. So don't worry too much. I'll be pointing out what you need to know and what you don't need to know. This is just for your own benefit. So, as I said before, catabolism provides the building blocks and the energy for anabolism. So catabolic reactions, represented on the top here, are generally hydrolytic reactions. So these are actions that use water to break chemical bonds, and they are exergonic or exergonic, meaning they produce more energy than they consume. Here you can see the glucose, which is a sugar molecule, that is broken down into carbon dioxide and water, and energy is released because this is an exergonic reaction. On the flip side of this, anabolism, anabolic processes often involve dehydration synthesis reactions. So these are reactions that will release water. I'll show you a bit better detail on this in a second here. And they are energonic, meaning they consume more energy than they produce. So here's the ATP being consumed. The amino acids are then being synthesized into larger protein molecules. So moving on to enzymes. So what are enzymes? These substances that I keep talking about. So enzymes are essentially substances that speed up a chemical reaction without being permanently altered themselves. So they're a form of catalysts, but they're specifically biological catalysts. Just a quick little tip here. If something has the suffix of ase, it's usually added to enzymes. So if you look at something like cellulase, that's an enzyme that breaks down cellulose. And RNA polymerase is an enzyme that synthesizes RNA. So that ACE is a good tip for you to look for, and then to identify the enzymes present in the processes. So what are the fundamental properties of enzymes? First of all, they have to be catalytic. They have to catalyze reactions. They have to be specific. They have to be composed of protein, and they have to remain unchanged during the reaction. That's very important. And then the location of your activity will determine how these enzymes are classified. Endoenzymes, endo meaning inside, are intracellular. Extracellular, exo meaning outside, are exoenzymes. So touching briefly on this specificity that we spoke about. So where is this the specificity of the enzymes stem from their structures? Think of this as similar to a jigsaw puzzle, where a specific piece will have a specific other piece that fits into it. That's very similar to how the enzyme and substrates function. So essentially, in this diagram, if we can quickly run through this, the enzyme represented by the blue will be contacted by a substrate, which is red, and this substrate will then move onto the active site that's indicated by where the arrow is pointing to. When it's attached to this active site, it will form the enzyme substrate complex. And then the substrate will be transformed into its products, and the products will be released. And very importantly, the enzyme will exit the process looking exactly the same as it did at the start. So it can be reused constantly. This cycle can be run constantly using the same enzyme molecule. Or sorry, not molecule, the same enzyme protein. So just in this example, this is an example of how the substrate is broken down, but just keep in mind that this process can look different also. Other transformations can occur. So moving on to the hydrolysis, as I said earlier, so hydrolysis is where forms part of many catabolic reactions, where you have a large molecule that is cleaved open by water into its smaller component molecules. And then you can see why this H2O will matter here, because the H2O will enter the molecule and break the bonds, and this is where your energy will come from. So this is the maltose that is being converted into glucose and glucose. And water here, you can think of it as lost during the reaction because it's consumed by the reaction. You can see where it ends up. And this is part of catabolic reactions. So this, you can think of this as energy production. On the flip side of this, you have dehydration synthesis, where you are removing water from two smaller molecules to create a longer or a bigger molecule. This forms part of anabolic reactions because they generally use dehydration synthesis. So if you look at these reactions as a quick review, dehydration synthesis used in anabolism, you have dehydration occurring, so the molecules are losing water, so they're becoming dehydrated. Not really, but you can think of it that way. And the synthesis portion is you're making a larger molecule, where hydrolysis, lysis means to split or to break apart. So here you have water being used, consumed, to break apart a larger molecule, and this will produce energy, and this will consume energy. So why do we care? Why do enzymes really matter? So enzymes decrease the required activation energy of reactions. So if you look in the diagram over here, you have a reactant called AB, right? And without the enzyme, your activation energy level is all the way up here. But with enzymes, you only need that level of energy for this for this reaction to take place. So thus more molecules of the reactant AB will be converted into the end product of A and B, because more of the molecules of the reactant AB will possess the required activation energy. So you're essentially decreasing the height requirement for the ride to take place, if you want to look at it through that little metaphor. But you are essentially decreasing the amount of energy required for this split from AB to A and B to take place. You need less energy for that to happen now because you have enzymes. And this increase is not small, as enzymes are extremely efficient. Under optimal conditions, they can catalyze reactions at rates up to 10 to the 8th or 10 to the 10th time. So up to 10 billion times higher than those, comparable to reactions without enzymes. So enzymes make a massive difference. That's why they're so important. So again, enzymes, what are they? They are, chemically, enzymes are globular proteins. So there are these protein molecules, and these proteins can either be simple or conjugated proteins. A conjugated protein are complex proteins such as hemoglobin, consisting of amino acids combined with other substances. And some of these enzymes require the addition of a non-protein molecule to function as an enzyme. These are known as cofactors. And without these enzymes, they will remain in the inactive apoenzyme form. So the cofactors can either be inorganic, it's like metal ions, or they can be they can be organic, such as vitamins or vitamin-derived molecules. And then if the cofactor is an organic molecule, it is called a coenzyme. You can see it being called a coenzyme there. And very importantly here at the bottom, I typically don't like to read all the slides, but this is pretty important, that the general characteristics of enzymes are the same for all forms of life. Okay, so that entire long-winded explanation I just gave you, what does that all mean? So essentially, coenzymes will activate your apoenzyme, creating a hollow enzyme or a whole functioning enzyme. So this little diagram really, summarizes it quite well, where your substrate will not be able to connect there because the shape isn't correct. So it does, the apoenzyme, the non-functional, you can almost think of it as a pre-enzyme almost, requires the addition of this coenzyme for it to activate. And the coenzyme can assist the enzyme by accepting atoms removed from the substrate or donating atoms required by the substrate. And some of these coenzymes can thus act as electron carriers, removing electrons from the substrate and donating them to other molecules in subsequent reactions. So just running through this again, E plus S here represents your enzyme, E is your enzyme, S is your substrate, E and S is your enzyme substrate complex, and then E plus P is your P is your products. So first you have your enzymes, your substrate will then attach to your enzyme, and then the reaction will take place, creating your enzyme product complex, and then the reaction will conclude with the products being expelled and the enzyme returning to its normal functional state before the reaction takes place. So this and that looks exactly the same, and then the reaction will just keep going, keep going, keep going, keep going. Yeah, like I said, the surface, the reaction will take place because the substrate has a high chemical affinity for the active site. So that's what also breeds you the specificity, is that the shape of the active site and the substrate molecules. Okay. And as I said before, the enzyme is very, very efficient. The turnover number is massive. I mean, 1 to 10,000 molecules per second. So this entire process of enzyme substrate complex, products being expelled, enzyme being ready again, enzyme being used again, is so quick. It can be up to 10,000 molecules per second that can be running through this. So it's really quick. Here's just another a diagram showing you what that looks like, or a 3D representation of what that looks like. And the shape of your active site here is where you are breeding some of your specificity, your active site. Look at the specific shape of your substrate and the specific shape of your active site, and that's where your specificity essentially comes in. So what can affect the activity of enzymes? Well, there are quite a few things. First, we're going to be looking at the concentration of substrates, pH, and temperature, and then we'll get back to inhibitors in a bit here. So first, looking at temperature. The rate of most chemical reactions increases as temperature increases, because molecules move more slowly at lower temperatures and may not have enough energy to cause the chemical reaction. For enzymatic reactions, however, elevation beyond a certain temperature, known as the optimal temperature, drastically reduces the rate of reaction. So here you can see the temperature, the rate of the activity will, or reaction will increase until a certain maximum. This is typically this optimal temperature, typically somewhere between 35 and 40 degrees Celsius. Think back to mesophiles. That might give you a tip as to why that is. After that, it will just decrease completely. And that decreases because beyond that optimal temperature, the enzyme will start to denature. It will lose its characteristic three-dimensional structure, the kind of the tertiary configuration. And the denaturation of that protein, of the enzyme protein, will involve the breaking of the hydrogen bonds and other non-covalent bonds. A nice example here that you guys are all familiar with is the transformation of an uncooked egg white, which is a protein called albumin, to a hardened state if by heat, so when you cook it. And that denaturation of an enzyme changes the arrangement of amino acids in the active site, altering its shape and causing the enzyme to lose its catalytic ability. Remember, I told you how important that active site is. So too much heat will cause it to lose the shape needed for the substrate to actually bind there. Just as a note, that heat isn't the only way that enzymes can be denatured. They can also be denatured by acids, as we will discuss here in a second, bases, heavy metals, alcohol, and ultraviolet radiation. So as I said, moving on to acids, or pH at least. So most enzymes have an optimum pH at which their activity is characteristically maximal. Here you can see again, enzyme activity will increase up to a certain pH and then it will just drop off. Above or below that pH value, which is somewhere over there between the 4 and the 6, so any pH lower than this optimal pH will result in decreased enzyme activity, and so the same is true for more basic conditions. So too acidic, not good; too basic, not good either. Essentially, why that is you have your H+ concentration, which is how you determine your pH in the medium. When that changes drastically, the proteins, again, the three-dimensional structure is altered, so the protein will become denatured. And this is what that denaturation will look like. So you can see here, this protein has a very specific characteristic three-dimensional shape. This is not an accident. This is what it's supposed to look like. And then either through heat or acidic conditions or basic conditions, it'll lose its structure. It won't look the way it should anymore, and thus it'll lose its function. This protein here, this denatured protein, is completely non-functional. Doesn't have any purpose. So now we will also be looking at substrate concentration, and here you would think more substrates, more reaction, but that is not really the case. There is a maximum rate at which a certain amount of enzyme can catalyze a specific reaction. So to reach the maximum rate, extremely high substrate concentrations are required. However, once you hit that level, further increase in substrate concentration on the x-axis here will not result in increased enzyme activity. It'll kind of plateau out. So under conditions of a high substrate concentration, the enzyme is said to be in saturation, and meaning that its active site is always occupied by substrate or product molecules. In this condition, a further increase in the substrate concentration will obviously then not affect the reaction rate because there are no more active sites that can be used. Just as a little caveat to this, under normal cellular conditions, enzymes are not saturated with substrates. At any given time, many of the enzyme molecules are inactive because there's a lack of substrate. Thus, the substrate concentration is likely to influence the rate of reaction because you have this, you're essentially starved for substrate, not starved for active sites. Ooh, here we go. So now we will be looking at inhibitors, the other manner in which you can affect enzyme activity. We have looked at some inhibition and how that works in the previous lecture. We're going to go into a bit more detail this time around. The last lecture was a bit rushed, and I feel I might not have discussed everything because there was just so much to get through. So first of all, there are two types of inhibition. You have competitive inhibition, and then you have non-competitive inhibition. So competitive inhibition is where the active site of the enzyme is filled by a competitive inhibitor, which then competes with the normal substrate for that active site. So here you can see your enzyme, and here is your substrate. And this is during normal binding. There's no competition here. There's no inhibition here. So that'll just bind and the reaction will take place. Now, with a competitive inhibitor, you have competition for the active site. This inhibitor will take the place of the substrate and stop the enzyme from reacting with the substrate. Thus, the reaction will not happen. Importantly, the inhibitor will not undergo any reaction to form products. So it'll just stay there. And that means that some competitive inhibitors can bind irreversibly to amino acids, preventing any further interactions with the substrate. Others can bind reversibly, and they can then essentially alternatively occupying and leave the action site, slowing the enzyme's reactions with the substrate under certain conditions. Increasing the substrate concentration can overcome reversible competitive inhibition, of course, because if you have more active sites, but not an increase in inhibitors, if you have an increase in substrate, they will start out-competing the inhibitors, essentially. So just to give a bit of a flashback to our previous lecture, remember we discussed PABA and the sulfur drugs, and that is a good example of competitive inhibition. So PABA or PABA is an essential nutrient used by many bacteria in the synthesis of folic acid. That functions as and it's a vitamin that functions as a coenzyme. When the sulfur drugs are administered to the bacteria, the enzyme that normally converts PABA to folic acid combines instead with sulfur anilamide, and the folic acid is then not synthesized, and the bacteria can then thus not grow. Then we move on to non-competitive inhibition. So non-competitive inhibition are essentially just inhibitors that do not compete with the substrate for the enzyme's active site. They just bind to another part of the enzyme. And this is represented here. Here again on the left, we have the normal reaction, no inhibition, active or competitive or non-competitive is happening. And here you can see the non-competitive inhibitor binding to another site called the allosteric site, which then alters the active site of the enzyme and prevents the substrate from binding. Again, this reaction can be either reversible or irreversible, but depending on whether the active site can return to its original shape or not. And that brings us to feedback inhibition. I remember how I said that allosteric inhibitor, that is the non-competitive inhibitors, can bind and create a reversible inhibition, essentially. And that's why they are used in a process called feedback inhibition or end product inhibition. So essentially, this is a control mechanism that stops the cell from making more of a substance than it needs, and prevents the waste of chemical resources. So in many anabolic pathways, the final product, here represented by these little dots in the diagram, so here the reaction takes place and it produces that little dot, that little final product. But now that final product in many instances can act as a non-competitive inhibitor, meaning if you produce this end product in enough of an abundance, it'll start stopping the reaction. It'll start interfering with the enzyme activity, thus preventing preventing the cell from making more of the end products than it needs. So how are we, why do we go through metabolism? And one of the major reasons is, of course, for energy production. And we get those from the molecular bond energy, which can be endergonic, remember, which would be to form a bond, or it can be exergonic, which is released when the bond is broken. Think back to when we discussed catabolic and anabolic reactions. And chemical reactions essentially then involve the transfer of these involve the transfer of energy in a series of many, and there's a series of many chemical reactions within a cell, which can be endergonic, energy absorbing reactions. So essentially these are reactions that cost the system, or it can be exergonic reactions. These are energy releasing reactions. They pay the system. So with that in mind, this diagram should look very familiar. I think when we started off at this, remember when I said catabolic reactions are exergonic, meaning they produce energy, and anabolic reactions are endergonic. They consume energy, they use energy. So catabolism pays the system, anabolism costs the system. And this is why enzymes are also so essential, because they conserve some of the bond energy in at least a biologically useful form. It makes sure that everything isn't lost as heat. So if you think of energy-rich chemical bonds broken in one step, think of something like lighting gasoline on fire. It's a lot of energy, but no biological organism can use the energy being released there. Because it's being released as heat. So the question then is, do living cells function on thermal energy? And the answer is no, of course not. So living cells require chemical energy. And thus most chemical bonds are not directly useful to the cell. So the cell must use a series of enzymatic reactions to produce a type of bond energy that is useful to the cell. Specifically producing ADP and ATP. This is adenosine diphosphate and adenosine triphosphate, and that is universally true for all living organisms that we are aware of. They all use ATP as an energy source. And the high energy bond of ATP is available for immediate use. It's essentially, you can think of it as the money or the currency that the cell can then spend at any time. Be it a little bacteria that's just moving around and requires energy for that, can use ATP for that. Or if it's you getting up and grabbing a coffee from Starbucks, the energy to do that is all ATP that you are using to drive all the reactions required to do that. Okay. So why is ATP so useful? So ATP has ribose and adenine, and then very importantly, it has 3 phosphate groups. These phosphate groups, you can see here, that little squiggly line over there, they represent a very, very high energy reactive bond. Thus, when ATP is broken down or converted to ADP, so triphosphate, meaning it has 3 phosphates, and diphosphate, so when it loses one of its phosphate groups, there's a large amount of chemical energy that is released, and then that energy can be used in other chemical reactions. That's what makes ATP so useful. So ATP is made through dehydration synthesis, and it is broken by hydrolysis to liberate the useful energy for the cell. So here you can see the adenosine triphosphate and water that is then converted into adenosine diphosphate, an inorganic phosphate, and energy separately. All these three things are separately, so that's not one bond. And that reaction, if it takes place in the direction from the bottom to the top, that would be dehydration synthesis. And if it takes place from the top to the bottom, that would be hydrolysis, because that's how you're liberating the energy that you need. So that is what the reaction looks like. And that is essentially what the metabolic goal of the cell is, because it facilitates the growth. It facilitates growth via making and spending of ATP. That's really what it all boils down to. That's what all the potatoes are about, or all the money is about. Maybe that's a better phrase. You guys might be more familiar with. And then, so once you have this ATP, this highly energetic and valuable ATP molecule, how would you harvest it? How do the microorganisms grab the energy that they now need from this molecule? And the answer of that is through reduction oxidation reactions or redox reactions. So yeah, guys, brace yourselves. We're going to start talking about some redox here. I'm sure many of you are aware of this process. So I'm hoping this wouldn't be too familiar or alien for you guys. So what are oxidation reduction reactions? Essentially, it's a type of biochemical reaction that produces energy that can be captured by the cell. And specifically, this is a very old little acronym, this oil rig, where oxidation is loss and reduction is gain. This is specifically of electrons. So you can see from the sketch here, oxidation, this is where you have an electron that is being lost. It's moving here. And reduction is where you gain an atom. So notably, the oxidation, this removal of this electron, is the reaction that produces energy. So the oxidation is where you get your energy from. Okay, so biological systems contain oxidation systems working together in a sequence to carry out metabolic reactions from a highly reduced energy source. So what does that mean? Essentially, a highly reduced energy source implies that more energy can be attained through its full oxidation. So it has the highest free energy. So if you look at methane, for example, it has a lot of energy. There's still a lot of energy that can be obtained from oxidizing it all the way down to carbon dioxide, which does not have a lot of energy in it. You can't really oxidize that anymore. So methane oxidation is a microbial metabolic process for energy generation and carbon assimilation from methane that is carried out specifically by a group of bacteria known as methanotrophs. And then this entire process can just be summarized by methane that is oxidized with molecular oxygen to produce carbon dioxide. Note specifically the presence of oxygen here. But yeah, so just think of this as that's a highly energy-rich molecule that the microbes want to use and break down and oxidize to produce energy. So again, as a review, very important, don't get this confused. Oxidation is the removal of electrons, whereas reduction is the gain of electrons. And then a redox reaction is an oxidation reaction paired with a reduction reaction. So here you can see the oxidation portion of it is essentially A losing its electron, so A is oxidized, and B that then is given an electron is reduced. But once it happens together, when A is given its electron to B to form A is oxidized and B is reduced, then this entire thing is known as a redox reaction. So something that you do have to note though, I just described it using electrons, that was just for simplicity's sake. In biological systems, the electrons are often associated with a proton because meaning together they are equivalent to one hydrogen atom. And these biological oxidations are often dehydrogenation. So what does that mean? A dehydrogenation is a chemical reaction that involves the removal of hydrogen, usually from an organic molecule. So specifically, another enzyme that you're going to, or coenzyme electron carrier that you're going to have to become very familiar with is NAD+. So NAD+ is nicotinamide adenine dinucleotide, just NAD+ is fine, is a coenzyme or molecule found in all living cells, and it plays a vital role in energy metabolism and maintaining proper cell function. It is particularly crucial for the functioning of mitochondria, which, what is the mitochondria? The powerhouse of the cell. And NAD+ essentially carries electrons from one reaction to another. So here you can see the H+, so the proton, but more importantly, the electrons it carries are transferred to the NAD+, which then acts as an electron carrier because it becomes a reduced electron character. And this organic molecule, because it now lost its electrons, has now become oxidized. It has undergone oxidation, and the NAD+ has undergone reduction. So this is exactly the same diagram as we looked in before. It's just that the H pluses are being, the hydrogens are being transferred, not just the electrons themselves. So in this case, NAD+ is an oxidizing agent. It accepts electrons from other molecules and it itself becomes reduced. And this forms NADH, which can then be used as a reducing agent to donate electrons. So this is how the cell can essentially manipulate where the electrons go. So that boils down to the question, what if life is a redox reaction? And that is pretty much true, because redox reactions, I mean, they're literally everywhere. A few examples from your everyday life that you're definitely aware of, like photosynthesis, the decay of organic matter, respiration, many biological processes, corrosion and rusting, combustion, and even batteries are all forms of redox reactions. So continuing on with oxidation reduction reactions, as I've stated before, many metabolic processes involve the oxidation reduction reactions because they need that electron transfer. And the electron carriers are often used to transfer electrons from an electron donor to an electron acceptor. So that gets us to this transfer of electrons. What drives that? Well, first of all, it can result in the release of energy, which can be conserved and used to form ATP, with more electrons being more energy rich, or if a molecule has more electrons, it is considered more energy rich. And then chemical species, this is really important because this will definitely be looking at this, and if you further your studies, this will become very important, especially in microbiology, the field of medicine. So I'd really recommend take the time, read up on this chapter, make sure you understand this. You need that strong baseline. So chemical species will differ in their reduction or redox potential. So redox potential is just a measure of the tendency of the chemical species to acquire electrons and thus be reduced. Reduction potential is measured in volts or millivolts, more commonly millivolts. And each species has its own intrinsic reduction potential. With the more positive the potential, the greater the species' affinity for electrons and density to be reduced. That is such a mouthful, but essentially what it boils down to is that your redox potential tells you how eager the molecule is to get rid of an electron or to keep its electron. That's essentially what it measures. It's a very baseline definition, but you have the full formal definition there, but it boils down to how difficult it would be to either force the molecule to take an electron or to take an electron from the molecule. So that boils down to the more electron negative molecule is, the better of an electron donor it is. So it has a weak affinity for electrons. It doesn't want electrons. Think of something like hydrogen. And the more positive your redox potential is, the better of an electron acceptor it is. So thus, it has a strong affinity for electrons. It wants the electrons. Think of something like oxygen. And the reason for that is, at least for I always found it much easier to envision this thing by using a periodic table. So ask yourself, what essentially would result in less effort to reach a similar state to noble gases? So what I mean by that is, what would be easier to reach to form that stable octet, that valence shell of 8 electrons? What would be easier to get to that state? So asking yourself that question will get you to the answer that the more valence electrons an element has, the more likely it is to gain electrons to form that stable octet. And the inverse of that is also true. The less valence electrons an atom has, the least likely it will be to gain electrons. It will much rather just get rid of its electrons, move down to a lower energy level, essentially. So thus your electron affinity will increase for groups from the left to the right across the periodic table. Think of something, again, like use hydrogen as an example. It has so much work to do if it were to gain electrons to get to 8 electrons in its valence shell. I mean, hydrogen not the best example because it wouldn't go to 8 because of how electrons are shaped, but just it would take more for it to do that than just simply to give up an electron. Whereas oxygen is only 2 away from having that 8 in its valence shell. So it's much easier to gain 2 than to give away what it already has. And then you can also, electron negative affinity will also increase upwards, because as the electrons are placed closer to the nucleus, they are experiencing that inward pull. Remember, your nucleus is positively charged because you have protons and neutrons in your nucleus. So you have to account for this shielding effect that is caused by the other electrons that are already orbiting your nucleus. So as you go down the periodic table, the shielding effect will increase. Thus, the repulsion will start occurring between the electrons. Essentially, these electrons are moving further and further and further away. Think of your orbitals around your nucleus. And these electrons are moving further and further and further away, so that the pull force between the nucleus and that far away electrons are much, much less than between the close electrons and the nucleus. I want to try and explain it like that. And then you also have those electrons that are already in the orbitals essentially pushing away against these electrons. They're pushing back against these newly added electrons because similar charges will repel each other. So you have that shielding effect where they're being pushed away. Thus, your electron affinity will increase if you move upwards in the periodic table. There are exceptions to this, so don't think of this as a fix-all thing. You're just going to have to study and know the exceptions. So these exceptions, you can kind of think of them as having an electron affinity of 0. Depends on the textbook, but most textbooks I've come across at least have reported them as having an affinity of 0. That is something like the noble gases. They're completely inert. They don't want to react. They already have their optimum state. Something similar occurs if you look at something like beryllium, Be, it's over here, which would require the addition of a new higher p orbital for the electron. I'm assuming as you guys keep going on chemistry, you'll understand a bit more what I mean by that, by your s and p orbitals. But essentially, it would require the addition of a new orbital, which requires a lot of energy to do. So giving the beryllium an electron would actually cost you energy. It wouldn't be easy to do. That's why they just say that it doesn't have any affinity, and its electron affinity is essentially 0. So here, then, this is our final slide for today. We have the difference between the electron negativity or the electron affinity of the donor and the acceptor. The greater that difference is, the more the energy that can be harvested. So your most energy-rich reactions will take place far apart on this diagram where you have a very, very good electron donor that's being paired with a very, very good electron acceptor, you'll have more energy being released, so it'll be more beneficial for the microbe to utilize that type of reaction if it's available. Okay, let's do some review questions here. Review question 1 is the factors capable of influencing enzyme activity are temperature, pH, substrate concentration, inhibitors, or all of the above. I'm going to give you guys about 5 seconds here. Okay, the answer, of course, here is all of the above. True or false? Catabolic reactions are generally hydrolytic. So are catabolic reactions hydrolytic? The answer, of course, here is true. So true or false? Atoms with a low electron affinity are more likely to lose electrons than gain electrons. I'll give you guys about 5 seconds for this one. And the answer here is true. Now, for a 5 more question, a longer question, so I'll give you guys a bit more time for this one. As you move down a group of the periodic table, does the electron affinity increase or decrease? Motivate your answer. So again, remember what I said before, where I want you to think of almost like 1 mark a question. There should be a small little paragraph that you're going to write. I'll give you guys about 10 seconds for this. Please pause the video if you really want to write your paragraph before looking at it, seeing how well you do. So I did provide you guys with a full written answer this time. I'm not going to read through it. It's literally just what I explained. So please take the time to review that. Make sure you understand why this is the case, and that is what your answer should more or less look like. It should contain all that information. It doesn't obviously have to be word for word the same, but the information should be the same. Everything I say here should be contained in your answer. Okay, guys, that's it for today. Again, just as a reminder, there will not be class on Tuesday, as you guys will be writing your tests. So I will see you guys on Thursday then for the next lecture. I'll see you guys. I will post the next lecture for Thursday then. Okay, guys, cheers.